Let's welcome to the stage Professor Rita Perlingeiro, who is the Lilly High Professor, Stem Cell and Regenerative Cardiovascular Medicine at the University of Minnesota, to look at the use of stem cells.
Well, I would like to thank you, the organizers, for the invitation to speak today. I have to thank them because I think I can answer at least the first question: what are stem cells? And the challenges, I think I can go on some of the challenges that have been talked about before me are in common, and there are some additional challenges for cell therapy as well. But in the next 20 minutes, I'll try to illuminate a little bit on the misconception and try to clarify what are stem cells, what the cell types that are out there, and what are the challenges, what they can do and what they cannot do. Hope I can do that. I need to disclose it's just one. I am a co-founder of Myogenica. It's a biotech startup that licensed the technology of all the technology we have developed in my lab for the last 20 years. So I don't need to really explain too much here, but you know the pathogenesis of DMD and the potential interventions, anti-inflammatories, antifibrotic, to try to circumvent the secondary issues and obviously gene therapy, exon skipping, to replace the missing dystrophin. What the cell therapy does? Cell therapy is another venue of potential therapeutic modality that you're going to be improving the regeneration that is defective in this disease, providing healthy regeneration. But as you provide healthy cells, these cells are also providing dystrophin. So how do you do that? So I just want to kind of keep a glimpse on when you're talking about cell therapies, there are two main types of cell therapy in terms of modality. You have allogeneic cell therapy, as this cartoon is showing. You have one donor. These healthy cells can be used for a significant number of patients. And if you're talking about Duchenne muscular dystrophies or muscular dystrophies in general, we are talking about one cell type that could be used for any patient with muscle wasting disorder. So advantages of that, it would be cost and accessibility to a large number of patients if these cells are effective. The downside of that is that you could imagine you need immunosuppression because it wouldn't be necessarily a perfect match. So it's not the cells from the patient. So this is something to keep in mind. In the autologous setting, the patient is the donor. So this is very nice, it's very specific. So the patients are generated, the cells are generated from the patient and injected back. So in terms of rejection, think about cell rejection because the cells are from the patient, but we still might need immunosuppression because of the protein that's being introduced like dystrophin that has not been seen by the patient before. So cons, well, it's very nice that it's patient-specific, but it can be costly and also takes time because in the case of Duchenne muscular dystrophy, the cells have to be genetically corrected before transplantation. So two main things that I want to already disclose from the beginning then is redosing. It's not a problem as long as the patients have been receiving immunosuppression, or in the case of autologous, potentially not. There is no significant issues, at least that we know, different from gene therapy. So there is no worry at the moment, at least, that the cells cannot be re-injected. So the patient can come back to the clinic to receive cells in different muscles. Immunosuppression, yes, in the case of allogeneic, autologous dependent, and we need to understand more these aspects depending on levels of mismatch, as you would have with a solid organ transplant. But let's talk about the stem cells and what type of cells are you talking about? There is so much hype about the stem cells and what are the cells exactly. So before we dive into that, I think it's very important to understand how the skeletal muscle regenerates and how this process happens. So here I have a healthy muscle. This is a mouse, but this is how we have pictures of, we are able to take pictures of this process which is really mimic in humans and other species. So when there is an injury, here is just an example of a myofiber, a cartoon, and then you are looking at the pictures of a muscle sample. So you have the myofiber here, you have the satellite cell in the periphery, which is the muscle stem cell. And when there is an injury here, these cells will proliferate and will expand, make myoblasts, they will fuse, and eventually generate new myofibers. But because these are stem cells, they also make more of themselves and go back to their resting position, waiting for another injury if necessary. So how does this look in a muscle biopsy, right? So this is a muscle, you know, in steady state, and nothing is happening, no injury. So you see these little dots here in the periphery. This is the satellite, the nuclei, is because there is no injury here. This is not a newly formed myofiber. So after the injury, you can see it became highly disorganized. It's because the cells, the stem cells have been activated, and the muscle is working. The stem cells are working to make new muscle to fix the damage. And as you can see, by 7 days, pretty quickly, you already see these new fibers. They are smaller, but they are pretty nice. And you can see the nuclei here. Now it's in the center. This is an evidence of a newly formed fiber. You see still some cells in the around, but by 2 weeks, it's pretty normal, back to normal. So the muscle has fully regenerated, and the only evidence, if you look at the muscle, that actually regeneration occurred, that the stem cells worked, is the position of the nuclei. So the muscle stem cells, they work very efficiently in healthy conditions. So what happens in DMD? So this is an example again in a biopsy that's unaffected. You see again the nuclei in the periphery. Pretty nice, not a lot going on in between the fibers. This is an example of an early stage biopsy. As you can see, there are nice myofibers, and there are also some fibers with the nuclei in the centers, showing that the stem cells of this muscle are working to compensate this damage because of the absence of dystrophin, and you see a lot of inflammatory cells. So the cells are working, but at the end of the day, they are still making myofibers that don't have dystrophin. So the issue, the damage doesn't resolve, as we saw in the previous slide. That is a continuous degeneration, regeneration. The stem cells keep working, but they are still making myofibers that are deficient in dystrophin. So the process is a continuous cycle, and it doesn't resolve. And as you can see here, by the end, after numerous cycles, you don't really have any more healthy fibers, and the tissue has been replaced by fibrosis and fat acid deposition. So one line of investigation that you can see that can help improve the pathology is to replace this tissue with healthy myofibers, but ideally also with cells that can provide myofibers, but also cells that can make replenish muscle stem cells. Because as you transplant cells here, you also have the patient cells making myofibers without dystrophin. So you want to also deliver stem cells that at long term will be making myofibers, healthy myofibers with dystrophin long term. So ideal cell therapy will be able to make both myofibers and provide also muscle stem cells to the tissue. So this is just a cartoon to visualize, very schematic. So you see the tissue sample, but you have here healthy myofibers, the nuclei is eccentric, but I just want you to appreciate that the muscle is not just fibers, right? So there are a lot other cells here that we have immune cells like macrophages, we have pericytes, the satellite cells here are in the periphery of the myofibers, as you can see. We have endothelial cells, and you also have some extracellular matrix, but minimum in healthy individuals. In the damaged muscle, you see you have the dystrophin-deficient myofibers, and they are leaky, and they are degenerating, and all this process of degeneration and regeneration brings changes to the muscle environment. As you can appreciate here, there is a significant number of inflammatory cells. You also have FAPs, fatty deposition inflammatory cells, which, as you know, there are several interventions trying to minimize these secondary effects that are very detrimental. So what you like to envision is, in an ideal cell therapy, is that this is the disease muscle, and when you have cells that will provide myofibers, the muscle stem cells, some of these fibers in the muscle will be replaced by healthy myofibers. Not going to be 100%. We're still going to have some of the host fibers, but this will eventually, hopefully, be taken over by new myofibers that will be generated by the muscle stem cells that were delivered. This would be the ideal scenario of a cell therapy, a stem cell therapy. So again, emphasizing that you want cells that will give healthy myofibers and healthy stem cells. So lots of misunderstandings and challenges. I'll try to cover some of them, right? So the liver is a big elephant in the room, so I don't even know if I'll be able to get there. But as you can imagine, muscles occupy a large number of our bodies. So as we start, we think locally, but it's always a challenge to target as many muscles as possible, and how many cells you need to target as many muscles. But let's talk about what are these stem cells and what cell types have been tested in the clinic, and the challenges and advantages, challenges, misconceptions. Okay, so again, I'm not going to repeat this one. I have already mentioned the importance of providing both cells that can make both myofibers and muscle stem cells. So what cells can do that, right? That's the important question. So you can have the right cells, but do we have enough of those cells? So this is two important points that I'm going to talk today. The modality we talked in the beginning, I switched the slide to the beginning, autologous, allogeneic, they both have pros and cons. Redosing is possible, and immunosuppression is likely necessary, especially to begin with both allogeneic and autologous. But let's talk more about what are the cells that you talk about, potential clinical trials in different locations and what's happening. So I'm not really going to go in details. We are way behind in terms of advances in clinical trials, but I will give you a sense of the cells that have been tested or are in plans to be tested, and where we are and what are the advantages and disadvantages or challenges, I would say. So this is the muscle hierarchy. We all want to generate myofibers, right? So we want a cell type in this muscle hierarchy that's going to give rise to the fiber. This is already the multinucleated myotube, so you really want a cell upstream. So these are the myoblasts. These are the stem cells. These arrows saying they can make more of themselves than they can make myoblasts, right? So what have been tested in the clinic? So the first one that was tested many years ago in the late 80s, basically in the early 90s, most of them, were based on exciting work in mice, but went very fast to clinical trials, and the results were quite mixed, but I say, overall consensus of these studies in Duchenne muscular dystrophy is the conclusion of these early clinical trials is that myoblasts, because it's downstream in the differentiation path, have limited regeneration potential. They are not stem cells. They are the daughters of the stem cell. They have limited migration and poor survival. So when these studies from these early clinical trials, this was the conclusion, that the cells didn't migrate much in the muscle. I have to say there are controversial studies, and I think one of the issues of these early studies is that the detection of dystrophy and the studies were all different muscles, different ways of culturing the myoblasts, but I think more important really the detection. So it's unclear if those studies that actually show a positive outcome, if it was really real or not in the case of DMD. Nevertheless, there are still clinical trials with myoblasts. They have been focusing more, not so much for DMD, but other applications that actually you don't need a highly regenerative cell, that you can inject the more differentiated cell and they do the job, but more localized, like urinary or sphincter incontinence. So not as much for genetic muscle wasting diseases. Okay, so really leaves us with the, I think from what I have talked in the beginning about the background is really stem cells would be the key population to be transplanted, right? So the satellite cells, the muscle stem cells, are the cells in the muscle that are responsible for regeneration. So if you can provide healthy satellite cells into the DMD muscle, these cells have the potential to regenerate fully into healthy muscle. What are the challenges? There are always challenges with the advantages, right? The challenges is that the frequency of a stem cell in a tissue is low. So being a muscle, a solid tissue, you have a significant biopsy to get enough muscle stem cells. So there has been enough studies, in other tissues, for example, hematopoietic, this is the bone marrow, you can somewhat expand, or collecting a large number of cells is not such a significant issue. So one way to try, which several investigators are trying to address this issue of low frequency, is try to expand the cells in the dish. So one issue with adult stem cells is that once you put them in the dish, they like to divide and make daughter cells. The daughter cells are the myoblasts. So it's okay if you have some myoblasts, but you would like the vast majority of the cells to be actually the stem cells, because these are the ones that will regenerate. So there are a number of studies trying to find strategies to differentiate the cells in the dish, or maintain the satellite cells, or expand it a bit and transplant. So in that sense, there are some phase 1 clinical trials going on in Simone Spuller's lab in Germany, trying to test what she called satellite cells outgrow, or muscle stem cell outgrowth. So it's not a satellite cell population pure expanded in the dish, but they have a technology that expands, they collect the tissue, and they maintain this for a few days, and they transplanted this outgrowth. So these are still very early, but there is hope with this population. But because of these issues, investigators have been trying to find other alternatives for stem cell therapy. So these are the cells you might have heard around. So there are the mesoangioblasts, or pericytes, work conducted by Giulio Cosso. There are the famous pluripotent stem cells that have the capacity to generate all cell types in the body. So as you can imagine, advantage, disadvantage to that. I'm going to go there in a minute, since this is my area of research. And there is a lot of talk about, when you hear about stem cell tourism, usually it relates to this, bone marrow and umbilical cord blood. And I have to be straight in the face that this don't make muscle. Okay, so if there is this pamphlet to go to Thailand to be infused with your own cells and you're going to get cured, it's not happening. Because the cells from the bone marrow or the cord blood, they can just make blood. Okay, so it might have a placebo effect, but they can not definitely make muscle. Okay, so I'm just going to get that out of the way. So going to the alternatives that have potential are currently being studied. Mesoangioblasts and pericytes, as I mentioned, these are also adult cells. They are present in the muscle, as I indicated here. This is a cartoon from Giulio Cosso. They are located here. These cells, when put it in the dish, they can expand. So they, different from satellites, they have the capacity to expand to a certain extent, again, because they are adult, but several studies from Giulio have shown that the cells can be, they have high potential for expansion, and they have been tested in different mouse models and also the dystrophic dog model. And these have evolved to clinical trials. There was an allogeneic trial published a few years ago. They were shown to be safe, but the efficacy was limited, so Giulio decided to move into an autologous therapy. So if I'm not mistaken, it's combining mesoangioblast with exon skipping, and this trial is currently enrolling. So this is happening at the moment. And then pluripotent stem cells, as I mentioned, there are these cells that have unlimited proliferation ability. So theoretically, they are great for genetic modification and expansion. They can make all the cell types in the dish, but as you can imagine, because they can make everything, you need to make sure you can limited for skeletal muscle. So they are first studied in the context of or in the human context of pluripotency with embryonic stem cells, right? Derived from the inner cell mass of the embryo, highly controversial, but that's how they got started in 1998 with Jim Thompson's paper in Science. And less than 10 years later, as you can imagine, many investigators want to find different ways to generate a pluripotent stem cell that's not embryonic, doesn't have all these ethical concerns. And Yamanaka managed to do that. Shinya Yamanaka, who got a Nobel Prize in Medicine in 2012 for his discovery, this technology of reprogramming that you actually can generate pluripotent stem cells from any cell type. In this case, you can use cord blood, bone marrow, but they're not going to go to make muscle. First, you have to make an induced pluripotent stem cell. Here is an example of using human adult fibroblasts, but can be any cell type of the body. With this cocktail of factors, using specific conditions, sounds like magic, but a magic that works, I have to say. In the beginning, everyone was a little bit suspicious about this technology, but it definitely works. That's why he got the Nobel Prize in 2012. And these are called induced pluripotent stem cells, because different from embryonic stem cells, they are not naturally, right? They are somatic, they don't do anything. But after you modify these cells, you reprogram, they become pluripotent. That's the name. So my lab has been investigated. Just want to give you an example of what could be generated from these cells. So my lab has been studied for a long time the capacity of the cells. Can you make just skeletal muscle, right? So we started with mouse to mouse in human cells, and they have robust expansion in the dish. We have transplanted these cells in different animal models, including dystrophin-deficient animal models, and look for my engraftment, as you can see here. And I just want to emphasize, I'm showing you work from my lab, but different labs have been using different strategies to make skeletal muscle from pluripotent stem cells. So this is an active area of research. So we have shown muscle grafts, and they also located under the basal lamina in the satellite cell location, so suggesting the cells give rise to muscle stem cells. So we have produced, we had done a lot of work, and you want to know if it actually was possible to generate, because we always talk about these cells can be generated large numbers, but we never really generate large numbers because we don't need to. You just need some millions for transplantation, right? So with support from Duchenne UK, we started this enterprise of applying a clinical grade technology to generate cells for therapeutic applications. And this is our cartoon. So they have pluripotent stem cells. We induce differentiation, and you induce PAX7 expression, which is the gene that's very important for the myogenic specification, and you generated these large numbers of myogenic progenitors really that can make both. So it's possible to generate very large numbers that can be banked, and so this is an allogeneic cell type. This is an example, okay? There are other labs generating other products, allogeneic and autologous. So you generated the induced pluripotent stem cells from a healthy donor. In this case, it's a cord blood sample. So cord blood can be used, but not for the final product, but for these induced pluripotent stem cells. We have then is a master cell bank, and I'm not going to go into details, but we have more than 100 vials of the cells in the freezer that were used for all the preclinical work and can be used for clinical trials all over the world for different patients or different types of muscular dystrophy or DMD, different mutations. So we have optimized the manufacturing protocol. This is clinical grade. We have shown efficacy in dystrophic mouse models, and this, everyone testing a cell therapy has to do this. This is an example of all the preclinical steps that are involved. I'm using an example of what we did because I know closer. And you have to show long-term engraftment, and more importantly at this stage, safety. So efficacy, we transplanted the cells in a mouse model of Duchenne. We transplanted directly in the leg. So to start it, this has to be local injections, right? So we tested the safety locally and you see human dystrophin in these mice, and MNAC, another human marker, and you see improvement in the muscle that was transplanted with cells. But really what the FDA wants to see is safety, right? So the maximum number you can inject feasibly in a mouse, and if you see the cells going outside the transplanted muscle, and if you can see any toxicity. So this is what we did. We transplanted as many millions as possible, which is not a lot, in a mouse muscle. And we collected organs, and we look for the presence of the human cells in these organs, and they don't go off target. They stay where they were transplanted. I was not very happy with the maximum feasible number that you could inject in the mouse, because as I mentioned, it's just a few millions because of the size, right? And if you're going to a human muscle, that is exponentially bigger. So we collaborated, and we perform studies in non-human primates using the same muscle that will be targeted in the clinical trial, the phase 1. And we show again that the cells that we transplant are able to make human-derived fibers. We also see human stem cells. A little bit trickier because the monkey is very similar to us, so a lot of the markers cross-react, but we managed. And again, we performed safety studies, and here we inject up to 67 million cells into one particular muscle and didn't see adverse effects, which is important. So after all this work that I summarize in a very few slides, a phase 1 trial was approved by the FDA, and Peter Kang is leading this effort. And as was mentioned before me, you have to be very specific with your goals, right? And this is a phase 1. We are very, knowledge of that you don't want to set yourself too very high expectations. The main one is really determine the safety. We have seen safety in animals, but the cell safety in the patients. Determine the maximal tolerable dose. Of course, you want to see engraftment. You want to see if the cells are there to justify for us to go in phase 2 and potentially future trials. And of course, as a secondary endpoint, see immune responses, because this is also another elephant in the room that we needed to be mindful when performing cell transplantation. So I'll end here. I'll be happy to take questions. I'd like to thank the group, all the funding agencies, the collaborators. These cells don't make heart, but sometimes we see one in the dish.